We've made a lot of different genetic modifications at the University of Missouri, and here's kind of a list. There's about 45 there. We've made over 1,000 cloned pigs. Lots of different applications, most of them biomedical, because there's lots of money in biomedicine to do these things. Seems to be there, at least in the past, there's been less money in agriculture. So I'm going to just focus on a couple molecules today that I have highlighted here in red for disease resistance. You know, we talked about earlier, we saw the slide where this costs $664 million a year in North America. And that's a good number. And if you look at Europe, it's probably, at least the estimates I've seen, it's 1.5 billion euros. You add all that up together, and that's $6 million a day in North America and Europe that this disease is costing. If those numbers are right, that's what it is. And we think of this as an economic problem, and yes it is, but this is an animal welfare issue. And we need to start thinking, at least I think we need to start thinking more about this as an animal welfare issue. And so the outline of my talk is I'm going to talk about a model for infection of PRRS. And I apologize for those of you that have been here the past few years, because a lot of these introductory slides are going to be the same as what you've seen before. But then we're going to finish up with CD163, some really exciting results there, and then we're going to have some timelines. So we had this model that's been developed for PRRS infectivity, and if I can get this to work. Yeah, so the idea is that first the PRRS virus has this loose association with heparin sulfate. It then binds the N-terminal region of Slaloadhesin. It gets internalized, and then domain 5 of CD163 actually uncoats the virus and facilitates the infection. And Van Breeden drew a really nice cartoon here that describes it. And this really sets the stage so that you can start coming up with hypotheses to test. Okay, the model is we first have that association with heparin sulfate, binds to the N-terminal region of Slaloadhesin, gets internalized into a vesicle, the pH drops, CD163 uncoats the viral genome, we get infection. Okay, with that we can then come up with a hypothesis. Is Slaloadhesin important? Well, let's knock out Slaloadhesin and see what happens. So we designed we designed a targeting vector here. On the top we have the Slaloadhesin gene. Exon 2 and 3 is where the start codons are. We pulled out the first few bases at the end of exon 1 and then basically deleted exon 2 and exon 3 where the start codons are, put in 3 stops going both ways so we're sure we'd get this thing knocked out. And then we did some transfections. Oh, incidentally, we included the neomycin selection cassette in there. Just as a side note, the FDA has already approved the product of the neomycin resistant gene as a food additive. So could we eat these? I don't know. So we did transfection, and we screened over 2,000 colonies. We found one targeted clone. The efficiencies on that are 7 times 10 to the 6th. Didn't work very well, but it did work, and we were able to take that clone and then do somatic cell nuclear transfer. And here's where we take an unfertilized egg. We remove the genetic material from that egg. We then take our donor cell that's been genetically modified. We place it underneath the zona pellucida with the unfertilized egg, fuse those cells together, deposit the nucleus into the cytoplasm of the egg. Hopefully the cytoplasm does its magic and reprograms that nucleus, and we end up with a blastocyst stage embryo that we can transfer to a surrogate to make a pig. And that procedure did work. Our pigs were first born in January of 2011, and these are heterozygote pigs for the Slaloadhesin knockout. Well, we had boys. We needed to make boys and girls so we could get homozygous knockouts, and that took a while. But our first Slaloadhesin pigs, and this is taken from the security camera actually in the farrowing room where the piglets were born, we had 4 that were homozygous knockouts, 4 that were heterozygous animals, and 4 that were wild type. And when I asked Bob, how many animals do you need? He says, boy, it'd be really nice to have 15, Randy. I said, well, let's shoot the rocket and let's see what we can get. And so we shot the rocket and we sent the pigs to Kansas State. After a week of acclimation, they used a low passage PRRS isolate, KSO6. It was introduced both intramuscularly and intranasally. Blood samples were taken on 0 through 35. And they were assessed for clinical signs. Pigs were co-housed. Again, no, no apparent clinical signs before infection. After day 3, they started to exhibit these mild respiratory signs. That resolved within a couple weeks, except in one pig. This is pretty typical for what happens to PRRS. The pigs were euthanized at the end of the study, did histology on the lungs. They all had severe to moderate multifocal interstitial pneumonia. We collected alveolar macrophages, did flow cytometry on the macrophages, and here's what we have in terms of a background, a Slaloadhesin in a wild type and CD163 in a wild type pig. And then when you look at our knockout pigs, here's what we have in terms of the Slaloadhesin peak. It basically moves back. It's superimposed over the secondary antibody, the background control. There's no Slaloadhesin being expressed on the surface of these macrophages. And if you look at all the pigs, we have the wild type pigs here. They all have 2 peaks. The heterozygotes all have 2 peaks. But the knockout pigs, they just have the 1 peak of CD163. So pretty sure we got it knocked out. Unfortunately, when we looked for virus, the cytopathic effect, the response was identical in all 3 genotypes. When we then looked at viral RNA within the blood, again, the response was identical through all 3 genotypes. So we conceived the idea actually back in 2002. I filed a disclosure with the University of Missouri at that time, and then we went looking for money, and we weren't very successful. One round at USDA, we got in the do not fund category. I imagine there's somebody else out in the audience that can join me in that, because there's other people that got in that category. But then we were able to find some money, scrape it together, produce a founder heterozygotes in January of 11, and the challenge began in November of 12. Publication in 13. This is an 11-year project. This took a long time, but we did get it done. So conclusions, well, Slaloadhesin knockout pigs, it's not lethal. We have pigs that run around. They seem perfectly healthy. The knockout mice do have subtle changes in their T and B cell populations and reduced immunoglobulin M. The Slaloadhesin pigs that we have may have an altered IgG response, but we really don't have enough to conclude that, enough animals, enough observations. There is still quite a bit of variation there. And the most important conclusion, the knockout did not affect susceptibility to PRRS. And again, if somebody wants access to these pigs, they're going real cheap. So I'm glad somebody laughed. So then we moved to the case for CD163, and I think Jay Calvert's done a great job in promoting CD163 as a candidate. I'm not going to go through all the experiments that promote that. I am going to cover a Van Gorp paper. But the question is still there. What molecule is involved in PRRS infectivity? A review that I read from just this past summer or fall, they're still talking about tetraspanin and CD151. They're still talking about vimentin. They're still talking about CD209. What is the molecule? We still have all these candidates out there. Why can't we go in and start to test for some of these? So CD163 is a member of a scavenger receptor cysteine-rich superfamily. And there's some evidence that if we look at exon 7 that codes for domain 5, that's really what's important. And I'll go through some of the information on that here very shortly. So Van Gorp did a really nice study where they tried to determine which of these extracellular domains of CD163 is involved in infectivity. And we have them numbered from 1 through 9 with a PST1 domain and a PST2 domain. And they took some hex cells and then they transfected them first with Slaloadhesin, because we know Slaloadhesin is important for infectivity, right? Well, maybe not. They then inoculated cells with PRRS and waited 24 hours and fixed them, and then they counted the number of infected cells. Now, I'm going to take a little bit of a sidebar, and this really isn't, and I apologize if this is meant if this feels as a jab at somebody, and it certainly isn't meant that way. But as I look at this, and I look at those PST domains, it's labeled that, but I'm not understanding. This is the amino acid sequence for CD163, and I've alternated colors for exons just to make it an exciting slide. But here's PST1 domain, and that thing, I only PST proline, serine, threonine. I only see 2 prolines. I see about 10 serines, and I see 1 threonine. That's a PST domain. And if we look at number 2, that doesn't even have a proline in it. It's got 3 serines and a threonine. I hope somebody afterwards or during the questions will explain to me why it was named that. So as I said, CD163 is a member of a gene family, and there's also a human equivalent. There's another molecule called CD163-like, and it has 12 domains. And if the numbering started at the other end, domain 5 would be the same in both of them. But in this case, domain 5 and domain 8 are actually the same domains. And what Van Gorp wanted to do was swap out some of these domains. And what they did was to swap out the domains and look at infectivity. And you can see here, if they swapped out domain 8, they dropped infectivity down to 0. And also in this group where they just did domain 8 rather than the 7, 8, 9, they knocked out infectivity. And so this provides some evidence that maybe just that one domain is what's responsible for the infection process. So we wanted to pursue this a couple different ways. One way was to use a traditional knockout by homologous recombination and just add a stop codon and knock this thing out. We also wanted to pursue the idea of a domain swap because we thought, well, maybe this thing's necessary for life and we really need to be careful with this. So some of the advantages of doing the domain swap, well, we know that SRCR, well, we think SRCR3 is necessary for hapten hemoglobin clearance. We know that domain 2, there's erythroblast binding. We know that domains 1 through 4 and 6 through 9, we have this tweak receptor. Bacteria are cleared by number 2. Well, that's probably a good thing to keep. And we think number 5 is responsible for PRRS virus binding or uncoating and/or uncoating. But is it really necessary? Well, hapten's ability to promote binding of hemoglobin to CD163 in humans has really high affinity, but not really so much in mice. In mice, CD163 accounts for only a small part of the hemoglobin clearance. And when they make these CD163 knockout mice, their conclusion was, and I have italicized here, there was no apparent phenotype change and offspring were viable and fertile. So at least in the mouse, maybe it's not real important. So then came along a technology called CRISPRs, and we used that to genetically edit some of these cells, and so we did it a couple different ways. One was we injected it into zygotes, and another way is we tried to alter somatic cells and then do somatic cell nuclear transfer. And what the CRISPR-mediated gene editing does is basically it cuts the DNA, and then there's non-homologous injoining. And basically that means there's a few bases added or removed when it tries to heal itself. And this can result in just changing a handful of bases. Thus, potentially you can remove exons, you can remove domains of proteins, or knock out a gene. And if we think about the swine genome is what, 2.7 billion bases? That's in the build. There's probably more like 3 billion bases for stuff that's not in the build yet. And we alter a handful of bases, and somebody's going to say, well, you can't eat that? I got a hard time with that. So the timeline for creating the CD163 knockout pigs for the somatic cell nuclear transfer, remember I said there was like 11 years for Slaloadhesin? This took us 5.5 to 6 months. And most of that's gestation. Okay. So genetic engineering, somatic cells, if that's what you're going to use, it can take quite a while. Somatic cell nuclear transfer and zygote injection, that can take a week. Gestation, puberty, we've got some long timelines, but certainly surmountable timelines. So I show these pigs because some of these are edited pigs and some of them aren't. In fact, the only way you can tell them apart is look at their numbers. And I think this one's number 43, and you're going to see that one later. But can you tell that pig apart from any of the others? I don't think so. So these pigs were sent to Kansas State University. And we had 3 pigs that were predicted to be null, and I have their genotypes here. Pig number 43 had 7 base pair addition in exon 7 from the maternal allele. In fact, all the 3 pigs that we challenged had that same allele inherited. So their 7 base pairs changed in this allele totally. And that results, because it's not divisible by 3, that results in a frame shift. You end up with a stop codon. You get a knockout. From the paternal allele, we had a couple different alleles inherited. One was 2 base pair deletion in exon 7 that also had 377 base pairs removed from the preceding intron. And then the other allele just had an 11 base pair deletion in exon 7. So if we look at that pig on the bottom, number 40, it has a net change. Well, we have 7 added and 11 deleted. As 4 bases net as the net change out of what, 3 billion? So we had the pigs were sent over, again, basically the same program as I described earlier. The identity of these pigs was blinded to the group at Kansas State. They were allowed to acclimate. They were challenged in this case with NBSL 97. This is, as Bob described it, this is a hot isolate. These pigs were maintained in the same pen. So even if you didn't infect one sufficiently, he's going to cough on everybody else. Okay? So these pigs are in the same pen. They're monitored daily for clinical signs, blood collected day 0 to 35. We terminated at 35, did lung lavage, and isolated the lungs and tissues for histopathology. So the pigs got sent over there, and Bob left other people in charge, and he went to the beach in Florida. Okay, so he's sitting on the beach in Florida. Did you preview these? He sent me this email. He says, Randy, below are the results. Everyone busted their butts to get it done. Let us know if we got it right. Maureen had sent him an email that says pigs 40, 43, and 55 remained negative at 4 days. Bob, it took you 6 minutes to forward it to me. So, clinical signs. We just scored them whether they had a fever or not. We also scored them for respiratory response, and the scores range from 0 to 6. On the next slide, if they had any respiratory score at all, it counted as a positive. So if it's a 1, it really doesn't matter what the rest of the numbers are. If it was a 1, it got scored. Now, for members of the press, I need to say that there's a press embargo on the rest of this data. And if you get up and ask me questions, I'm going to have to very politely deny or decline answering it. This is in press at Nature Biotechnology, and you guys can see it next week, and then we can talk about it next week. Anybody else, they can ask questions, and I'll be happy to answer. So if we look at the clinical signs, here we have on the top panel the wild-type pigs. We get respiratory signs, over half the pigs by day 7, respiratory signs, it diminishes off. Same thing with the fever, comes up and it goes down. Those 3 wild-type pigs, there's nothing there. These pigs don't get a fever, they don't cough. They're in the same pen with these other pigs coughing on them. There's absolutely no sign. I think Bob now says, Randy, you don't need 15 pigs of a genotype. You need 1 pig of a specific genotype, and we can challenge these pigs and know if they're resistant to this or not. So the surface expression was determined, as I previously described, using flow cytometry, looked at viremia, looked at antibody response, did histopathology, pretty much standard procedures. And here's what we have for the viremia. Here's your wild-type pigs. Viremia comes up day 4, comes up pretty high. Pretty normal response. Here's the knockout pigs. There's nothing there, guys. It's zeros. Here's the antibody response. Comes up a little bit on day 7. By day 14, there's antibody. That's pretty normal for the wild-type pigs. These knockout pigs, there's nothing there. These pigs don't see this virus at all. Lung pathology, here on the left we have the wild-type pigs, and on the right the knockout pigs. Knockout pigs, perfectly normal lung pathology. The wild-type pigs obviously have interstitial edema. We've got some infiltration. This is a detailed list of the lung pathology, and you can read through this. The scores on the right are from Pat Halber's paper back in '95, and you can see a range of 1 to 4, and we have our 3 knockout pigs on the bottom. There's no lung pathology at all. These pigs are just perfectly normal. If we look at the expression of CD163 and 169, again, 169 is Slaloadhesin. If we look at our wild-type pigs, we've got CD163 expression normally, CD169 expression normally. Here we have our CD163 knockout pigs. Slaloadhesin, it's there, but the CD163 knockout pigs, the peaks equal to the background control. These are knockout pigs. There's no CD163 there. At least it was recognized by this antibody. So overall conclusions, Slaloadhesin pigs are not resistant to PRRS. CD163 knockout pigs are resistant to PRRS. We are working on challenging with other genotypes, and we are working with different isolates right now, and you're just going to have to stay tuned for some of those results. So we have this model, and this is really good. Somebody put up a model and said, this is what we think is happening, and now let's see if it's true. And so we went in, we knocked out Slaloadhesin. Maybe that's not the molecule involved. We knocked out CD163. It's definitely involved in PRRS infectivity. CD163 is a gatekeeper. So where are we going to go from here? Well, this technology is going to allow us to answer a lot of really neat questions about both basic and applied questions for biomedicine, but also domestic animal biology, and answer some really important questions for agriculture. The debate's been out there. Which molecule is involved? I think we now know CD163 is. So there's a lot of people that helped out with these projects. The paper that will be coming out next week, Dr. Kristen Whitworth, who's sitting back here, is the first author on it. Kevin Wells has been absolutely instrumental in helping design some of these CRISPRs and really pushing the molecular biology forward. A number of people involved in transfection and genotyping, zygote injections. I think XJ is going to see Kehoe Lee on the slide here. He was intimately involved with that. We've got another crew that's been involved with the nuclear transfer and embryo transfer. Some of you guys might actually know Clifton Murphy. He's 89 years old, and he still goes out and showers in and does our surgeries for us. We have a crew that takes care of the piglets. Melissa Samuels really heads that up, and everybody pitches in and helps out when they need to. The Slaloadhesin project was really pushed forward by John Green and Tina Egan. And then I have my collaborators at Kansas State University, Bob Rowland and his team that's listed here. And then Genus has helped fund us and been involved in some of the later work with CD163. Alan's really been involved. Dave McLaren, some of you might remember him. He used to be at Genus. He's been really involved, and then John's been involved. Funding sources for Slaloadhesin knockout, ARS actually helped out a little bit with that. Food for the 21st Century at the University of Missouri, the Christopher Columbus Foundation, and Genus have helped fund the CD163 project. And now I'm going to do a little shameless advertising. We have an international conference on pig reproduction a year from this summer. I hope you'll consider coming to the University of Missouri for that. And I do have an open postdoc position. I hope somebody would be interested in applying for that.